Mack, and this is the tryptophan operon. When the operon is off, tryptophan is absent, and the RNA polymerase is able to bind to the promoter and flow through the genes of the operon. This process is called transcription. Transcription of the tryptophan repressor produces an mRNA segment and translates into a protein repressor, which is inactive due to the absence of tryptophan. However, when the operon is off, tryptophan binds to an allosteric site and turns the repressor active. The active repressor then binds to the operator, blocking the RNA polymerase, and therefore no RNA is made. Black eye operon is naturally off due to it being negatively induced. Black eye is a regulatory gene that produces mRNA and translates into a protein. This protein becomes the active repressor, which comes over here and binds to the operator. When it binds to the operator, it blocks RNA polymerase from transcribing the 3 genes. This is what needs to happen because the 3 genes metabolize lactose. However, lactose is absent when the operon is off, and therefore it's only wasting energy. However, when lactose is present, lactose binds to the active repressor and causes it to change shape. When the repressor changes shape, it is no longer able to bind to the operator. Because it is no longer able to bind the operator, RNA polymerase can freely move to transcribe the 3 genes, LAC-C, LAC-Y, and LAC-A. LAC-Z codes for beta-galactosidase, which hydrolyzes lactose into glucose and galactose. LAC-Y codes for permease, which is a membrane protein that allows lactose to move into the cell. Lastly, LAC-A codes for transacetylase, which removes an acetyl group, therefore breaking it down. CRP uses the same as the LAC-I operon. There can either be high levels of CAMP and low levels of glucose, or high levels of glucose and the absence of CAMP. When there are high levels of CAMP, CAMP binds to the inactive CRP, an allosteric site on the inactive CRP, causes it to change shape and become active. The active CRP then binds to the RNA polymerase. The 2 proteins work together and they speed up the process of transcription. However, when there are no CAMP, nothing is able to bind to the CRP, therefore RNA polymerase still transcribes the genes, however it's just much slower. RNA splicing begins with pre-mRNA. Pre-mRNA consists of introns and exons. Exons are the expressed genes. Introns, however, are the non-coding segments. In making proteins, we don't need the non-coding segments, therefore we must rid ourselves of the introns. This is done using SNRPs. SNRPs are consisted of snRNA and proteins. Many SNRPs, along with other proteins, combine to make a spliceosome. Spliceosomes then attach themselves at specific splice sites located at the end of introns. Once they are attached, they cut the intron, immediately joining the exons. Then you are left with an exon segment and excised introns.